Welcome to the course on basic genetics. The teachers for this course are Dr. Richa Arya, Professor Madhu Gavaldas Tapadia, and myself, Jagat Kumar Roy. We are from Department of Zoology, Banaras Hindu University, Varanasi. This is week 2, lecture 2, regulation of gene activity in eukaryotes. Within this, initiation, elongation, and termination of transcription. There are 3 different RNA polymerases in eukaryotes. RNA polymerase 1, this is for ribosomal RNA transcription. RNA polymerase 2, this is for heterogeneous nuclear RNA transcription, or in other words, one can say mRNA transcription. RNA polymerase 3, this is for tRNA, 5S rRNA, and other small RNA transcriptions. Transcription factors are needed for RNA polymerases to bind with the promoter. RNA polymerase by itself cannot bind with the promoter. So, there are different proteins they are required, and they together bind with the promoter. If we say eukaryotic RNA polymerase, in prokaryote I am sure you had a knowledge of the RNA polymerase, where you have alpha subunit, beta subunit, beta prime subunit, and sigma factor. Eukaryotic RNA polymerase, it has some of the fragments, they are related to bacterial subunit beta prime, which binds with the DNA. That has C-terminal domain tail containing specific sequence. In yeast, the number of the amino acid of the tail is 26. In mouse, this is 52. So, this is one subunit. The other subunit, this is related to bacterial subunit beta that binds with the nucleotides. Another subunit which is less than 50 kD, and this is related to bacterial subunit alpha. There are some subunits in 30 to 20 to 30 kD range. They are common to all 3 polymerases, and there are certain which are specific for specific ones. So, let us first consider, because there are 3 RNA polymerases, there has to be difference in the initiation of transcription. So, let us first come initiation of transcription by RNA polymerase 1. For RNA polymerase, there is a core promoter and there is upstream binding region or upstream control element. That means this has 2 regions. One is core promoter, which is near the start site, with which the Tata binding factor and SL1, SL1 is one of the important transcription factor for RNA polymerase 1, this binds. So, core promoter is 1, and with the upstream control element, which is several bases up, and with this the upstream binding factor that binds. So, several upstream binding factor binds, and upstream control element is minus 110. So, this is minus 110 to minus 170, minus 110 to minus 170. The core promoter, this is minus 40 to plus 20. This is minus 40 minus 40 to plus 20. So this is the core promoter. What are the function of these two elements? The core promoter and the upstream control element. This is to bring more and more of RNA polymerase. With the upstream control element, upstream binding factor, they bind, and there is protein-protein interaction with upstream binding factor and the RNA polymerase. Thus, a large number of RNA polymerase come in this region. This is by protein-protein interaction. And with the core promoter, there are certain transcription factors which are there. SL1, this is 1, and TBP, Tata binding protein, which binds with that region from where the RNA polymerase will sit and start opening the DNA. So this is how the initiation of transcription occurs, that RNA polymerase comes, binds here, then moves and then transcribes the region. When initially the RNA polymerase is bound, that time the transcription starts. RNA polymerase has not moved. If RNA polymerase start moving, then that transcription continues. Otherwise, those transcripts which are formed when RNA polymerase is at stable condition, at halted condition, that will not go, those will get aborted. So that is aborted transcription. Ribosomal RNA transcriptions at nuclear organizing region. The question is where ribosomal RNA genes are present. Just now we talked that ribosomal RNA genes, they are transcribed by RNA polymerase. Where are they present? In the nucleus. They are present in the nuclear organizing region of the chromosome. There are certain chromosomes which are also known as satellite chromosome. There you have secondary constriction. So secondary constriction means primary constriction is the centromere. The secondary constriction, another constriction where the DNA is to some extent open up, and thus the other piece, the part of the chromosome, it appears as if disconnected or as if broken from the main. So that is why this is called as satellite, but actually it is not. That region which appears as if nothing is there, there you have the ribosomal RNA genes, and plenty of them are, that means many, they are in many copies. They are very active in transcription, and that is why that region is very much opened up. This gives the feeling as if there is Christmas tree. That means if this is the DNA and if this is transcribing, that means one RNA polymerase, it has gone from here to here. So that means it transcribed up to this. So this much part is transcribed. The next RNA polymerase might have reached here. So from here there is another. So this is first one, second one, third RNA polymerase has already reached there. So it has transcribed. The earlier RNA polymerase has reached further, so they transcribe like this. And that is how a Christmas tree-like structure is formed. So these are the transcripts which have got transcribed from here till here. This is the transcript, this is another transcript where it has not reached up to here but up to this. So same way you have this kind of a situation. So the same thing you can see here. and because I said there are several copies, so you have these several copies. This is one, this is another, this is another, this is another, like this. This is another. So there are several copies. So that means at a time, lot of ribosomal RNA is formed. One gene is being transcribed by a large number of RNA polymerase. Now, initiation of transcription by RNA polymerase II. The initiation is different. In RNA polymerase I, we have seen that there is core promoter, there is upstream control element where some of the factors bind that brings the RNA polymerase or attracts RNA polymerase. RNA polymerase binds with the TBP and SL1 is there. RNA polymerase comes with them and finally it starts transcription. RNA polymerase II transcription is slightly different. Here, this is the transcription start site, this is the transcription start site. There is a box called Tata box. This is a short element that is present. This is INR, close to INR, and then there are certain other boxes which are also there. So this constitutes the promoter. This promoter is the basal promoter. That means this much is required for any transcription. What binds first? First, the transcription factor 2D, that binds first. Where it binds? TF2D has TBP, Tata binding protein, and this is called Tata box. So that means TBP will bind with this. So TF2D is here. Then next in the series, there will be TF2A. So TF2A has come and bind. Then there will be TF2B. This is TF2B. And TF2F. So TF2F binds here. This is TF2F. In Tata-less promoter, the same transcription factors, they function. But TF2D, this is the first one, TF2D, this is with some other TAF, that is Tata associated factors, bind INR. So here the TF2D binds with the INR. Other transcription Tata associated factors in TF2D recognize DPN. DPN is on this site. DPN is on this site. So that is called as downstream promoter element. So if Tata box is not there, they are called as Tata-less promoter. There, this region plus some region in the downstream part becomes important. So DPN is in this region and INR means initiator, that is there. So this is the whole complex that is formed. Here the last TF2E and TF2H, they are required to melt DNA. TF2E is there, this is TF2E, and TF2H is there, TF2H that has come. They are required to melt DNA. When you say melt DNA, that means the DNA double helix or double stranded DNA will open up and will become single stranded. This TF2H, this has ATPase activity, this has helicase activity, and this has kinase activity also. Kinase activity, where kinase activity is required, this I did say that there is in RNA polymerase II, there is CTD tail. that means C-terminal domain tail. And this C-terminal domain tail, when phosphorylated, then only this RNA polymerase can go out, can move from here. And that means the promoter which was bound with the RNA polymerase 2 is now cleared. So, this is called promoter clearance. Now, initiation of transcription by RNA polymerase 3, I did say that there are different RNAs, they are transcribed. There has to be 5S rRNA, there are tRNAs, there are many other small RNAs. So, type 1, that is for 5S ribosomal RNA, there are two boxes, box A and box C. That is very important. And do you know where these boxes are located? This is transcription start site. These boxes are located in the downstream region, not in the upstream region. So, RNA polymerase 3 promoter is the downstream region promoter. For tRNAs and other RNAs, TF3B, TF3C and polymerase, they bind with box A and box B. In the first one we had box A and box C, here we have box A and box B. And both the box A and box B, the factor that binds, so they are important and they allow the RNA polymerase to come and bind. So, these are the example of downstream promoter. That means promoters band in the downstream region. Here also it is binding like this. But RNA polymerase will sit at +1 region. That means transcription will occur from here only. Okay. So, this is initiation of transcription by RNA polymerase 3. If we talk about elongation of transcription, this picture I showed in my previous lecture also, that this is already initiated and new bases are being added so that this chain grows. So, this is called as elongation. And then comes the termination. Termination of transcription is different for different genes. RNA polymerase 1 terminates transcription at an 18 base terminator sequence. There is a sequence called terminator sequence, and after, so then for RNA polymerase 3, that terminates transcription, usually a second U in a run of 4 U's. So, there are 4 U's. At the second U, the transcription termination occurs. Why U's are important? Because U will have double bond with A. And if this region is embedded in GC rich region. So, around you have GC rich region, and here in between you have the other one, that is AU. So, result is that they will get, because they have only 2 double bonds and there are no 3 bonds also. So, they are separated easily and thus the transcription is over. In RNA polymerase 2, 3' end is generated. RNA polymerase 1 we said that this terminates in the terminator. In RNA polymerase 3 also there is terminator, there is a specific region where transcription terminates. In RNA polymerase 2, transcription continues. One does not know clearly where the transcription termination will occur. But here what happens, that as the RNA is getting transcribed, there is a sequence AAU that is there. So here you can see that this is the RNA that is synthesized. This is the RNA. And with this, here you have AAU a sequence. With this, one of the factor binds. This another factor that binds in the other side, where there is a GU region and there is region which needs to be cut. So this is the nuclease which will be able to cut this. So now when this is cut, so 3' end is now generated. When 3' end is generated by this cut, then with some of the factors, there is poly U that is added. So a stretch of approximately 200 U's that are added. So that means at the end of mRNA, there will be poly A, because so many A's are being added. But it is not that all mRNAs are poly A plus. There are poly A minus also. So at this moment, I do not have a clear example that I can give you, but otherwise, this is the way the end is generated. So with this, we come to the activity and discussion. We said Tata box. This is TATAA. Is this a conserved sequence or this is a consensus sequence? We categorically said that Tata box is there, and with this the TBP, that is Tata binding factor, binds. But mind it, this is not a conserved sequence. This is not there in all the promoter exactly the same sequence. Here you can read that you have T80, A95, T45, A50, A50. That means this T, in 80% of the promoter, this T is there. In 95% promoter, this A is there. In 45% promoter, this T is there. In 50% promoter, this A is there. In 50%, this A is there. So this is a consensus that is generated by analyzing a large number of promoters. Then the next question, how the three polymerases recognize their specific transcription units? We will come to this. In RNA polymerase II transcription, where termination occurs. Termination is not at the region where we get the 3' end generated. Termination occurs maybe several bases away from there. So a clear terminator has not been very well categorized for generalization. There are specific terminators that is there, but for generalization, one is not having that. But certainly, there will be U stretch within a GC rich region. So wherever such U's are there, because there are only two hydrogen bonds, so they have a tendency to get broken. So with this, I stop here. Thank you.